Hello everyone and welcome to the webinar. Today I am Usman Ashraf, working as an application scientist for liquid handling here at FORMULATRIX. Today we are going to discuss about miniaturization and its benefits to maximize laboratory efficiency. Our talk will focus on the importance of miniaturization achieved by using our automated liquid dispensers, MANTIS and the TEMPEST. It will be highlighted by experimental findings from our valued customers. Please note that the webinar is being recorded and will be available on our website after the session today. Before we begin, if you have any questions during the presentation, please enter those into the chat box and we will try to answer as many of them as possible today. If there are any questions we are not able to get to, we will follow up after the presentation. And with that, I am going to begin with the presentation. Firstly, we will get to know about miniaturization and its possible benefits in scientific analysis. We can define miniaturization as scaling down the protocols normally used for a specific experiment. The question that comes into mind is why we need to miniaturize the protocols? And the answer lies in the benefits that we can obtain by miniaturizing our experiments. There are certain advantages of miniaturization, such that we can perform the analysis with lower sample volumes, which enables the efficient utilization of precious and limited reagents. For example, if we look at the figure, we can see that the amount of the analyte being used for catalyst screening assay decreased as the protocol got miniaturized. As we went from 96 well plate to 1536 well plate, we also increased sample density per plate, which is another benefit of miniaturization. This will help us increase the number of replicates for an experiment, leading to improved data quality and reproducibility. In the same run, miniaturization can enable narrowing down our analysis to a single cell. Most cell-based biological assays yield averaging data from large group of cells, but we can have more specific data from a single cell analysis with miniaturization. This approach is beneficial for studying omics at the single cell level, such as genomics, proteomics, and metabolomics. Another benefit associated with miniaturization is that it increases the throughput of the analysis due to less time on handling plates, so more samples can be processed at a given time. The miniaturization can also reduce running cost of the assay as work can be performed with low quantities of the reagents. Moreover, when coupled with automated tipless dispensers, miniaturization can cause a significant reduction in waste that we will discuss in the next slides. All these benefits of miniaturization can be effectively achieved by automation. The key requirements for miniaturization like low sample volume, precision, and accuracy can be fulfilled when coupled with automation. For that purpose, we have state-of-the-art automated liquid dispenser, MANTIS and the TEMPEST. Both instruments make use of non-contact microdiaphragm-based chips for dispensing liquids. MANTIS is a single channel with up to 48 inputs, while TEMPEST is up to 12 channels that contains 96 individually controlled dispensing nozzles that allows higher throughput. Tempest, being a multi-channel robot, has slightly higher CV and volume increment than the Mantis. The low dead volumes, that is 6 microliter in Mantis and 48 microliter in Tempest, can also reduce the cost by conserving precious reagents. Another key feature of these instruments is that they can handle highly viscous liquids up to 25 cp, which is equivalent to viscosity of 70% glycerol at room temperature. Let's have a look at the technology of these devices. The heart of the system is a flexible silicon diaphragm sandwiched between the two layers of the hard plastic and input at the top and output at the bottom. This precisely meters out aliquots of liquids from 100 nanoliter and above. The process is really simple. The chip has pneumatically activated diaphragms. When the Mantis picks that chip, it connects to the vacuum and pressure used to manipulate the two diaphragms. Vacuum is used to lift it up and pressure is used to push down the diaphragm when the input valve is open. The selected diaphragm will be filled with the liquid. Most importantly, this is a very precisely and accurately measured space. It can't be overfilled, and timing ensures that it can't be underfilled. Here is how the liquid is transferred from the reservoir to the target plate without using the tip while dispensing at the great speed. Now, I will share some examples of where these instruments were utilized for miniaturization and what benefits were achieved. Here is a miniaturized qPCR assay analyzing the expression profiling of retinal pigment epithelium cells in response to the applied drug. Usually, it takes almost 60 days to prepare an RPE monolayer from human stem cells, so the sample is highly precious. That's why miniaturization was required. After the drug treatment, cDNA synthesis and expression profiling with qPCR was performed by full-scale and 10 times miniaturized reaction. Here, Mantis was used to dispense the master mix in the miniaturized assay. When analyzed, very sharp amplification and melting curves were obtained even at miniaturized reaction of 2 microliter volume. In another experiment, the RNA-seq library was prepared at the full-scale reaction and 4-fold miniaturized scale and were compared. cDNA was synthesized directly from the cells as well as from the purified RNA. Full-scale and miniaturized library prep were performed from both samples. In a miniaturized assay, all the reagents were dispensed using Mantis, and it can be seen in the figure. The size distribution analysis of both assays produced similar results. Equivalent size fragments were observed in both reactions. Furthermore, cDNA product yield was also analyzed in both experiments. Here, the graph shows that average cDNA produced directly from the cells in full-scale and miniaturized reactions were comparable. Similar results were obtained when cDNA synthesized from pure RNA was analyzed. The number of reads from RNA-seq from cells and pure RNA were also equivalent as shown in the graph. Let's move to another example where MANTIS was used to miniaturize the HGRF assay. Homogeneous time-resolved fluorescence is a novel technology applied to many antibody-based assays to study kinases, biomarkers, protein-protein interactions, and other molecules of interest. By using conventional pipetters, the reaction consisted of 20 microliter in 384 well plate. That was reduced to 5 microliter in 1536 well plate when miniaturized. Here Mantis was used to dispense 0.5 microliter of the donor and acceptor antibodies into a 1536 well plate. The graph here shows the results of the analysis in which full-scale, two-fold, and four-fold miniaturization were done. When compared, all the assays produced signals of almost similar strength. Similarly, when the dispensed reproducibility of all three assays was quantified, very low coefficient of variation were observed. Apart from efficient and reproducible results, other benefits obtained by employing Mantis are summarized here. A significant reduction in plate number and consumable cost, specifically the tips, made this experiment cost-effective. Similarly, labor-intensive pipetting was also avoided, with less hands-on time at bench. And lastly, a lot of antibodies were saved, indicating the importance of Mantis and miniaturization for such kind of assays. Alpha screen is a bead-based homogeneous assay that does not require separation steps or washes. Full-scale and miniaturized assays were performed in an experiment to study the protein-protein interactions. The full-scale reaction was performed in a 384 well plate, while the 1536 well plate was used for miniaturization. Here, Mantis was used to dispense donor and acceptor beads. As can be seen, the reaction volume got reduced to 3 microliter from 25 microliter, along with the manifold reduction in the use of protein for entire screen. Moreover, this allowed them to assay approximately 300,000 data points with an equivalent amount of reagents through miniaturization. As far as the data quality is concerned, we do not have a comparison, but the work has been published and they screened 73,000 compounds, having a 9 hits coming out. Here is a scatter plot of the z-scores observed across 56 plates used in the primary screen. Z-score was also above 0.5, so the data quality is actually very good using this miniaturized methodology. Here is another experiment that was designed to improve the scalability of neuron-based phenotypic screening assays. Primary neurons remain important tools for in vitro neurobiological investigations, especially for drug discovery. They are large and extremely sensitive cells requiring additional care for handling, making the scalability of assay very challenging. The researchers at Scripps Research Institute were previously using 96 well plates for culturing neuron cells. They used to seed cells with an automated liquid handler. As they planned to miniaturize the assay for higher throughput, the liquid handler they were using was not compatible with 1536 well plate, so they switched to using MANTIS. After miniaturization, the cells were analyzed for growth and density. As shown here in figures, uniform cell growth and similar densities indicate comparable viability in all plate formats. Based on these results, we can infer that Mantis is gentle enough to dispense highly fragile and large neuron cells even for miniaturized experiments. In another experiment, the scientists employed Mantis to miniaturize the organoid culture of beneath cells, which are secretory cells of the small intestine. Conventionally, organoid culturing is performed manually in 48 well plate, which is both labor intensive as well as of low throughput. So, the researchers miniaturized their experiment to 384 well plates using benches. The organoids thus generated showed uniform distribution and morphology, as can be seen here in the figures. Different compounds were then screened, and a comparison was performed. The benefits obtained were significant. As using Mantis, 8x faster plating was done, 12x less reagent was used, and 10x greater number of samples were analyzed, rendering the process time efficient, cost effective, and of high throughput. Here we will discuss about use of the Tempest for screening assay on drop array plate. Working with drop array plate poses a challenge for non-contact dispensing as the plate do not have wells and we have to dispense at exact locations on the plate, avoiding cross-contamination and splashing. Interestingly, this wasn't a challenge for Tempest, as although being multi-channel, it dispensed the cells and reagent at specified locations in a 384 well drop array plate. The efficiency of the Tempest in combination with miniaturization is highlighted by the results obtained when the drop array plate was used for miniaturizing the screening assay for viability testing of HPRE cells. The standard screening assay required 40 microliter of reaction per well in 384 well plate. By using Tempest with drop array plate, a tenfold reduction in volume was achieved. However, similar results were observed when the seeded cells were analyzed by viability staining. Here is the overall summary of our talk today. We have discussed the benefits of using Mantis and Tempest in different miniaturization experiments. The experimental data indicates that our products can be utilized in a variety of applications such as PCR, alpha lysa, organoid culturing, and for dispensing a range of chemicals and reagents including antibodies, beads, and cell suspension. Similarly, their ability to dispense low volumes precisely can be utilized to develop miniaturized protocol without compromising the data quality. Moreover, Mantis and Tempest can reduce the experimental cost by saving the expensive reagents, as can be seen here, where 4 to 10-fold volumes of reagents were saved in various experiments. In the end, I want to show how FORMULATRIX is revolutionizing liquid handling. We will now talk about two new but very different liquid handlers, FAST and FlowI8. FAST, with its one-of-a-kind positive displacement, fixed 96 channels is specifically designed to transfer low volume liquids precisely. True positive displacement pipetting delivers exceptional accuracy at low volumes, irrespective of liquid class, which is crucial when setting up miniaturized reaction. As fast is liquid class agnostic, so there is no need to program liquid viscosities, making it good fit for non-aqueous liquid transfers. Some unique features of the instrument includes flexibility of picking 96, 12, 8 or 1 tip with positive displacement head, computer vision aided automatic tip rearrangement, and removable automatically calibrating deck. On the other hand, Flow I8 is an air displacement pipetting liquid handler. It has 8 independently spanning channels used to transfer wide volume range with great accuracy and precision. Flow I8 is compatible with variety of liquid types as it automatically optimizes pipetting parameters while dealing with volatile and viscous liquids. Its unique features include automatic deck calibration, labware and tip detection, tandem liquid level detection and tracking, and touch sensing. Here is the illustration of different pipetting technologies used in our liquid handlers. Fast positive displacement tip is a specialized disposable tip consisting of a capillary and a piston that operates like a syringe. The piston comes into direct contact with the liquid and draws it up and down within the tip, eliminating the air cushion, increasing precision and accuracy. The direct contact with sample results in the aspiration force remaining constant, which makes it easier to pipette highly viscous, volatile, foamy, or hazardous liquid samples. On the other hand, Flow I8 is an air displacement pipetting system more similar to handheld pipetter. A small air cushion exists between the piston and the liquid during the process. The piston inside the instrument moves down to let air out. This means the air is displaced by the piston and the volume of the air displaced is equivalent to the volume of the liquid aspirated. Flow I8 tips contains conductive and pressure sensors in it that detect the liquid levels within the tip and automatically optimize the pipetting parameters accordingly. This mitigates the effect of viscosity on air displacement pipetting, making Flow I8 highly precise and accurate. Now, let's have a look on FAST and Flow I8 in action. The FAST is a quick, reliable, and easy to use liquid transfer system designed to address any liquid transfer across life science applications. Its unique 96 tip head allows rapid sample transfer between 96 and 384 well microplate using 96, 12, 8 or 1 tips. With a positive displacement pipette tip, the precision of the fast liquid handler is not affected by the types of the liquid samples that are being transferred. It has a broad range of applications including NGS, enzymatic assays, antibody assays, and cell-based assays. It can also perform cherry picking, serial dilution, plate copy, and plate reformatting. The Flow I8 is a smart liquid handler, ideal for optimizing your protocols and bench work quickly and easily. It allows you to focus on research, science, innovations, and breakthroughs instead of programming complex parameters. It is the first of its kind that comes with the sense of touch. It senses the bottom of the well prior to dispensing a sample and then senses the side of the well for a tip touch without the prior definition of labware geometry. The Flow I8 liquid handler takes liquid level detection and tracking to the next level with sensors that enable dynamic liquid level tracking without prior input of the liquid properties. Pipetting accuracies and precisions are guaranteed without user adjustment of pipetting flow rate, equilibration time, or offsets of various liquid classes. So I just want to stop here because there is a lot more to come and it will continue. A little about our company. FORMULATRIX is a growing company that focuses on automation in life sciences. It was established in 2002 to provide protein crystallization automation solutions. Since then, we have started developing other laboratory automations, including next generation liquid handlers and filtration systems. We aim to enable you to formulate your scientific discoveries by making automation precise, reproducible, and fast. We have more than 4,500 instruments installed worldwide in pharmaceutical industries, research institutes, diagnostic and academic laboratories. Within our Formulatrix family, we have more than 1,000 employees from various fields such as engineering, biological sciences, marketing, designers, and media management worldwide. Okay, now I would like to answer some questions. And the first question that comes in is, we cannot hand pipe it down to a very low volumes as the error becomes too large with respect to the total volume. How does the Mantis addresses this? Okay, it is true that traditional handheld pipettes have a higher error at low volumes, especially below 2 microliter, but Mantis shows high accuracy and precision even at 100 milliliter, and that is due to its diaphragm-based microfluidic pump technology. The chip is pneumatically actuated with precisely metered diaphragm cavities to ensure reliable and repeatable performance. Another question comes in with regards to the return on investment. How can I calculate my cost saving when miniaturizing my reaction volumes? How can I justify the cost of the instrument in terms of ROI? The answer is, you can estimate your reagent cost saving by calculating the fold reduction in reaction volumes and expect additional savings on consumables such as microplates and tips. For Mantis, we have a ROI calculator available on Formulatrix website. That tool lets you input variables such as plate format, tip consumption, and working days to instantly calculate the time to recoup your investment. The Mantis often pays for itself in a matter of months. I hope that answers your question, but if not, just let me know and I can answer that in detail offline. Another question that comes through is, how do I validate that the reduced reaction volumes work as well as the recommended values from a kit manufacturer? Does Formulatrix partner with kit manufacturers to publish data showing that there is no detriment to data quality? So, when the specific direction is not available from the kit manufacturer, I think this can be validated by performing the miniaturized and full-scale reaction in parallel. This method will yield optimal reaction volume for a given experiment while maintaining data quality and integrity. To the next part of the question, yes, Formulatric has collaboration with some of the most well-known kit manufacturers. One we have mentioned in this webinar is Takara Bio. They have published a quarter volume protocol of their SmartSeq 4, a cDNA synthesis kit. Next question that came in, what workflows benefit most from reaction volume reduction? Do you have a list of workflows that have been tested on your instruments? So in this talk, we have summarized several workflows such as PCR, immunoassays, and cell-based assays that can be miniaturized according to assay sensitivity. However, single-cell analysis, NGS, and assay development would benefit the most from reaction volume reduction. For the list of workflows, we do not publish a master list, but you can contact us for assistance with your workflow. Our team will help provide to cost analysis and customer references. Another question is, other than cost savings, what are the benefits of reaction miniaturization? So a major benefit is that one can perform more replicates of an experiment, providing more data points to improve the data quality. It also helps to increase the throughput of the experiment as smaller volumes can better optimize in microplates. Moreover, miniaturized assay have become more automation friendly and total waste production is considerably reduced. So I think it covers all the questions that have come through. I will really appreciate any feedback that you have. I want to thank you for participating in the webinar and Q&A session. With that, we close this session. I hope you have great rest of your day. Thank you everyone.